So, new paper came out about how to detox from the spike protein in your body. This is spearheaded by Dr. Peter McCullough. I think that might be a recognizable name for some of you. And not long ago, I did a video on potential ways of detoxifying from spike protein injury. This is either post-infection or potentially post-vaccination. And I asked the audience, look, let me know what compound or treatment option you would be most interested in for me to do a video. And I tabulated the most common mentions in the comments. And the one that won was Natokinase. So, okay, as promised, I will do a video on Natokinase now, but I already did a video on Natokinase. So I thought on top of that, I would also do a video of a compound that I've never yet do a video on. And the first compound, so the top three was Nattokinase, N-acetylcysteine, and vitamin D, and I did video on all of these. So then the next in line, where I didn't do a video on yet, was Bromelain. And as it happened, just at the end of 2023, these authors published an article on both Nattokinase and Bromelain, as well as Curcumin, as an option to treat against spike protein. So then, perfect. I thought this is an excellent combination to do now a video on this topic. My name is Dr. McCullough of Genomics. Let's get started. So why even target the spike protein? So remember when the vaccines were being offered against COVID-19, it was mentioned that the spike protein itself would stay in the site of injection, would not last very long, and it was assumed as well that it's inert, that it will not cause any damage. Unfortunately, it appears, based on the slowly accumulating evidence, that none of these turn out to be true. And as a consequence, now we know that spike protein can't. And by the way, I'm not on skis anymore, so I could fall through snow at any moment. So we'll see if that happens or not. Just a fair warning. And so first of all, spike protein, it appears that it in itself can have toxic properties. So we know that it can cause inflammation, but it also appears to be causing unusual clots, and we did really big series on that topic, how spike protein can be contributing to clot formations, including it has been isolated from clots, isolated from people who had it. So check that out. So there's couple ways of mentioning how spike protein could be problematic, and it seems to be affecting many different biological systems. So metabolism can be affected, neurology, cardiology. So it's not inert. It definitely doesn't seem to be inert, and of course it also has profound immunological consequences. And we still have to figure out what all of these are. And then, in terms of where the spike protein goes post-vaccination, well, we don't really know that information. The best information actually comes from autopsies of people who were vaccinated, and based on that information, now we know that spike protein was found in cadavers in brain, liver, kidneys, ovaries, and testes. So definitely spread throughout the body. So that does not seem like it's staying in the site of injection. And then finally, clearly, spike protein has been established that spike protein can be around for a long time, both post-vaccination as well as infection. So recently I made a video showing that there at least that spike protein can last up to 6 months, even in some individuals post-vaccination, and long periods of time have been also observed post-infection. Post-infection, there have even been recorded instances for up to a year, so very long time. And therefore the authors of this paper proposing this treatment option mention, look, targeting spike protein directly might be a very valuable option as a consequence of all of these issues that we potentially are seeing with the spike protein. And degrading the spike protein could be very valuable. The authors propose that it's the lack of degradation, of breakdown of the spike protein, that they think might be contributing to its longevity, which I thought was very interesting, because clearly there's also scientific suggestions that there might be different cell reservoirs that act as cells that act as reservoirs for spike protein. And I made a video suggesting, for example, bacteria, right? And there's other cell lines as well. They didn't mention at all anything like that. So I thought it was interesting that they think it's because the lack of degradation of the spike protein oops, here I go, that might be causing why spike protein lasts so long. And they didn't provide any evidence for those statements as to why spike protein itself would not be necessarily easily degraded. But here we go. So then they suggested, they divided the paper into two chapters, if you will. One chapter was devoted to how we can degrade the spike protein, and that included both Natokinase and Bromelain, which we'll talk about in just a moment. And then the next chapter was on how to prevent negative function of spike protein remaining in the system, or the spike protein fragments. And they're proposing use of curcumin as well. So we're going to go over all of that. So let's get started with the degradation of the spike protein and nattokinase. So nattokinase, check out my previous video on this topic. Nattokinase comes from a specific bacteria called, I'm going to give it a try anyway. I'm pretty sure I'm going to say it wrong. Bacillus subtilis natto. It's a bacteria that ferments soybeans. And it's that bacteria that produces the nattokinase. And apparently, natto, the food made with fermented soybeans, have been and hence nattokinase has been used in Japanese culture for centuries as a potential form of medicine against anti-atherosclerotic purposes. So meaning for prevention of the formation of plaques in your blood vessels, as well as for anti-coagulatory purposes. So basically, to prevent formation of clots. So powerful stuff, obviously, right? So next we're gonna move on. Okay, cool. That's basically the little background. Next, so clearly those are some of the potential properties of nattokinase from a health or medical point of view. Next, we're going to move into what kind of evidence do we have of nattokinase against the spike protein, and that's the video I made where a group of scientists were showing that nattokinase could potentially destroy spike protein directly. And there's others who also shown that nattokinase could prevent virus infection of cells. And the one more evidence that I thought was really interesting was that nattokinase had been apparently shown that it could destroy the spike protein of infected cells. So I thought that was also really interesting. So, however, keep in mind, this is all in vitro, meaning it's against cell lines. None of that do we know what's happening in physiological systems in humans. But that's the evidence we have. All right, next we're going to move to dosage. So what kind of dosage are we talking about here? So that's some of the most common questions I was getting on that video. That's one of the reasons why I was so interested in this paper as well, because these doctors, medical doctors, provided some great summary of dosages. So apparently nattokinase has been tested for safety up to 80,000 fibrinolytic or fibrinolytic units. What does that mean? Basically, fibrinolytic means breakdown of fibrin, which is the component of clot formation. So when we're talking about fibrinolytic units, we're talking about how well something can break down clots. And 2,000 fibrinolytic units is equivalent to about 100 milligrams of nattokinase. And the authors also mentioned that with just 2,000 units, after you take 2,000 units of nattokinase, within mere hours, you'll be able to see clots degradation elements flow. You can isolate such elements of showing that clots are being degraded, such as D-dimers. D-dimers are basically leftover molecules after breakdown of clots. Check out my first video dedicated to how spike protein is involved in clotting, in order to get background information on this, on what D-dimers are, and how do we get them. So then you'll get some background, and other fibrin and fibrinogen breakdown components as well. So just mere hours after just 2,000 units. Okay, so then these authors are proposing this is definitely a good starting point. All right, and now for safety. How safe is nattokinase? So first of all, their pharmacokinetic and pharmacodynamic studies have never been done with nattokinase, so we know absolutely nothing with regards to that. Meaning, basically, how safe it is once it's floating in your blood, however, they do mention that nattokinase has been used a lot as a supplement, especially in recent times, and based on all that evidence so far, anecdotal evidence, it seems safe. But there are issues, and therefore you need to be careful. First of all, you could have allergic reaction for those who are allergic to soybeans, so you have to be careful with that. Another big one is the fact that, remember, it acts as against clot formation, so therefore it can lead to excessive bleeding. So therefore, if you are using nattokinase, you should be using with medical supervision, and especially if you are using anticoagulatory drugs or antiplatelet drugs that already cause increased potential bleeding issues, right? So very, very important that if you use nattokinase, and especially in conjunction with these medications, that you should be using this supplement under medical supervision. They stress this a lot in that review, in that paper. Okay, then we're now gonna move on to bromelain. So bromelain that's actually a name that, if I follow this correctly and I understand this correctly, encompasses number of proteolytic enzymes. So not just one. So and they're extracted from pineapple core. So what are proteolytic enzymes? Basically, we're talking about proteins that destroy other proteins. Okay, that's what that would mean. And again, bromelain is known to be anticoagulatory, so it can once again it can degrade clot formation. It's also if I remember correctly, anti-inflammatory, so it can modulate our immune system, and it has already been approved in Europe for medical use, and this is for cleaning of burn wounds, like wounds after being burnt. So I thought that was pretty cool, pretty interesting. And so that's the little background on bromelain. So what evidence do we have about bromelain and spike protein? So apparently it has been shown that bromelain can destroy spike protein as well. It can prevent infection of the virus by the virus of cell, infection by the virus of cells. So it can negate that. And I think, if I remember correctly, it might reduce expression, meaning production of the receptors needed for SARS-CoV-2 virus to infect our cells as well. So that's the evidence we have for bromelain against spike protein. Okay, so now let's go to dosage, and the standard dose of bromelain right now that is used in medicine, they were mentioning, is 500 milligrams. So again, this is the proposed starting dose. And now safety. Now, in terms of safety, bromelain, once again, remember, because it's anticoagulatory, so you have to be careful with excess bleeding. Once again, you need medical supervision. But also on top of that, it can cause now severe allergic reactions in some. So this is not trivial compound, and can also lead to gastrointestinal issues in some as well. So keep that in mind. And if I remember correctly, I think it was bromelain as well, that could affect function of number of different medications. So it can really influence whether the medications are working properly or not. Many medications, again, as a consequence, if you're thinking of bromelain, you really need to make sure you seek proper medical supervision. And some of these are very obvious medications, and the list was long. Like, that included even chemotherapy medications, opioids, antidepressants. So some standard medications. So keep that in mind as well. And once again, the authors call for use of medical supervision if you're going the route of using this compound, this supplement. All right, okay, we're going to move now to curcumin, which is the compound they mention to use to mitigate any negative functions of the spike protein. Curcumin, of course, is isolated from turmeric, and it's very well known for its antiviral properties, and it achieves that by reducing inflammation, by affecting how transcription factors are used in order to activate inflammatory responses. So basically, transcription factors are the little machinery robots that help to decode genetic information of the DNA inside a nucleus into mRNA, and then the mRNA is used as a template to produce proteins of interest. And in this case, it would be products that lead to inflammation. So this is how curcumin can work. So it can reduce inflammation caused by viral infections, and it's been shown to be antiviral against many different viruses. The list was long. I would not be able to even recall fraction of that, including SARS-CoV-2. So that's the little background on curcumin. Now, in terms of evidence of curcumin use against spike protein, here it's the weak sauce for sure. It's the weakest evidence, because the only evidence we have is from computational modeling. So in silico studies, meaning it's not real, it's all just done on a computer, and based on computational modeling, it seems that curcumin can bind to both spike protein as well as the ACE2 receptor, and therefore it could be blocking their interaction. But it's based on modeling only. I can tell you that on these computational modeling of molecular interactions, they are fairly powerful these days, and used extensively even in the development of drugs. You have to be. These models are actually very accurate. They're pretty powerful these days, but nevertheless, we don't have real evidence. So this is the weakest component of those three compounds for sure. Now, in terms of dosage, they mentioned that World Health Organization recommends up to 3 mg per kilogram of body weight, which they said on average should be about 250 mg per person. And then let's move on to safety, I guess. So in terms of safety, going over 250 mg per dosage could cause problems, as in it can lead to gastrointestinal issues, as as with bromelain, as well, including even causing ulcers. And curcumin can also affect proper function of certain hormonal therapies. So again, if anyone is using curcumin as a form of therapy and is in conjunction using such therapies, once again, the authors claim you need to be under proper medical supervision. So if you're using any of these, and even if your doctor wouldn't agree with you, I think it would be very wise to let your doctor know, so that the doctor can monitor what is happening to an individual clinically. So and that will be it. So as a consequence, now the authors propose that these three should be used in tandem, and we're going to talk about that in a moment. But they also mentioned other compounds. There was a long list of compounds that they didn't expand on. They just this was their list that they proposed that could also be considered for similar purposes as these top three. But on top of that, the one that was of special mention was the N-acetylcysteine, and we did a video on that compound as well. So please check it out. The reason why I bring it up is actually a couple of reasons. Number one is because they provide a dosage of what is the proper daily dosage of N-acetylcysteine, and they mention between 400 to 1,000 milligrams in a day. And that brings me to another paper that I wanted to interject into this very spot on bromelain. That was one of the papers I've studied before I came across this paper to make a video on. And this was an old paper from 2021, where the authors were suggesting of possibility of using both bromelain and acetylcysteine as a combination to help against COVID-19. And to briefly summarize the results of that paper, what their authors did is they basically used both bromelain or acetylcysteine, or a combination of both, to target spike protein, and they were able to show that acetylcysteine in the concentration that they used, which I believe was, I think they used 20 milligrams per mil, and then bromelain used 25 micrograms, 50 micrograms, 100 micrograms per milliliter. So they used different concentrations. The N-acetylcysteine in their hands did not destroy spike protein. Bromelain did, but as well as bromelain and acetylcysteine did as well in dose-dependent manner, meaning that the more was used, the more bromelain was used, the more destruction was observed of spike protein. So I thought that was interesting and worthy mentioning. Then they also tested both an acetylcysteine, bromelain, and the bromelain and acetylcysteine combination, which they, by the way, called Bromac. And they tested that to see how well it would stop the virus infection of specific cells. And in this case, neither I think it was neither acetylcysteine or bromelain were able to stop the infection, but a tandem combo was effective in stopping the infection of cells. And again, this was in a dose-dependent manner, and they were able to do this. How did they measure that, by the way? I was mentioning in one of the recent videos how you can measure behavior of virus, because remember, viruses are super tiny, and they're not easy to study. So one of the ways you can see is how well do they infect cells and kill the cells. Because of course, once a cell is infected, it will die eventually, because it will burst to release the viral progeny that were born inside the cell. So they were basically looking at viability of cells, how many cells are alive when you expose them to the virus. And if you add the compound, and the compound prevents the virus from killing the cells, you can see how well the cells can survive because of that, right? So that's what they did. And they also reconfirmed the bromelain and acetylcysteine combo, Bromac, that it was preventing infection by looking at how much virus genetic information was produced by the cells. And they simply use standard PCR, the same PCR technology or test that we all been through type of thing when during the pandemic. Basically, you look for specific genetic information of the virus, and you amplify that genetic information to see that it's there. So, and in that way, they were able to once again show that Bromac was preventing virus replication inside cells. So very, very interesting stuff. And the authors mentioned, look, we definitely should be considering this as a potential therapy option. Now, of course, they mentioned, look, these are caveats. These are just in vitro studies, meaning they might not be equivalent to what's happening inside a human body, right? And one of the examples they mentioned, and I thought this was really, really interesting, is that the pH of the body can change under viral infections, so that the mucosa of inside our nose is about pH of 6.5, and when a person gets infected, the viral infection can really change that up, even increase it up to pH of 8. And basically, which is a measure of acidity level. The lower the pH number, the more acidic the environment. The higher the pH number, the less acidic it is, or more basic. And depending on what the pH is, you can dramatically change the behavior, biological behavior of compounds in the system. And therefore, what you might see in vitro, meaning in a laboratory, in your little petri dish against cell lines, because of the pH changes, might literally not work in the human body. And therefore, they say, look, the only way we really need to know is we would have to do clinical trials, proper clinical trials, to truly know. Which brings me now back to the original paper I was discussing by Dr. Peter McCullough and others, and they reconfirmed, look, we don't have any evidence of how this works, of all these compounds that they mention as a potential therapy for what's happening in with long COVID and people suffering potential spike protein related injury. We don't have any physiological evidence, and whether this truly works in humans. The only evidence is what I mentioned, is so far how it works against spike protein. And they mentioned, when they looked at the time of writing of this article, whether such clinical trials might be happening, there aren't any, none whatsoever. So keep that in mind, that this is truly would be experimental. However, that's what they propose at the moment. And it's also a bit of an unusual way, because typically antiviral methods do not target a specific viral protein. Typically, what antiviral methods do is they block interaction between the virus and the host cell. So this would be a little bit different. So again, keep that in mind. This is all hypothetical. We don't have any evidence in terms of clinical trials at all, whether this truly works or not. These authors are just proposing it as an option, because obviously we're having a lot of issues in terms of post-pandemic problems, right? With and which we now are describing as long COVID. Nevertheless, what do they propose in terms of treatment? And they're proposing this for medical doctors, and they're proposing that one should be taking 2,000 fibrinolytic units of Nattokinase or 100 mg twice per day, as well in combination with 500 micrograms of Bromelain, also twice per day, as well as 500 micrograms of nano or liposomic curcumin. I did not mention that the 250 mg per mil, 250 milligrams was a curcumin dose that was recommended. But if you're using the nano or liposomic version of it, then you can increase that dose to 500 hey, 500 milligrams, and then you also increase the bioavailability of curcumin. And another side note is that if you don't use that one, then you can use the regular curcumin, and you can increase the bioavailability with a compound called, I think it was piperin or something like that. Either way, it's extracted from black pepper, which is which I thought was interesting because I've heard this long time ago. I never double checked this, but I've always, and I often cook with turmeric, and I always add black pepper as well to it in order to increase the bioavailability of turmeric. So, and I always was wondering if it's true or not, and now I know it is. So that I thought was really interesting. So they're mentioning also 500 milligrams of that nano or liposomic curcumin twice a day, and they recommend that people who are suffering because of the spike protein related injury, they should do it for at least at least 3 months, and even up to a year if needed. So basically, that's the summary of how much they're recommending this treatment. All right, so this basically brings me to the end of this. I thought this was really a cool way to give you the summary. Oh, I'm falling through. Give you the summary of the.
Hey, you're that YouTube guy.
Yes, I am. now you're in the YouTube video with me.
YouTube video. There you go.
Yeah.
Your buddy's waiting for you back at the hut.
Yeah, that's right. I'll be with him right away.
Cool. Have a good one. Happy New Year.
Same to you guys. And and where was I? Got a little distracted. Oh yeah, I thought this was a really cool way to give you a video on the most interested compounds that as you voted for. All right, so thank you everyone for your support. Please like, please share. As always, you know, this is how we grow. Please leave a comment. Let me know what you think about this treatment option. Especially would be interesting to know what doctors might be thinking of this. Obviously, I really wish we got clinical trials ongoing with this. We still have no official treatment of any kind for long COVID at all, and hence I think this is great that doctors are looking for solutions. I think that's very valuable. All right, and also please check out our Patreon account where we add additional content that we don't post on YouTube video, and I look forward to seeing you in a future installment. And Happy New Year. Bye, everyone.